Welcome. My name is Robby. I'm a senior at Cedar Shoals High School. I love to play sports. Right now I play tennis and I swim for Cedar, and I plan on going to college for mechanical engineering, hopefully at Georgia Tech. So where I work, my internship is based out of the College of Engineering, and our research is in the Regenerative Bioscience Center. So our location is in the Animal and Dairy Sciences building, which is right here. It's right next to Ramsey. So I work in Dr. Luke Mortensen's lab. Dr. Mortensen is an assistant professor at UGA, and he studies the body's potential to heal itself. My more direct advisor is Bobby. Bobby's a graduate student at UGA, and he's working towards his master's in engineering. So what we're going to do today is we're going to go over where I work and who I work with, what I study, the background of what I study, my research going forward, and what I've done so far. So what I study, the big picture is the potential to use mesenchymal stem cells in cell-based therapies. And so mesenchymal stem cells are a type of cell that have natural anti-inflammatory factors to them. So we want to try to use them to help chronic inflammatory diseases such as arthritis. So as we can see in this pretty picture here, we have some dermatitis, which is always inflamed, and so we hope to cure that inflammation or at least reduce it. So our bodies are made up of trillions of cells, and to be able to organize them into an organism, they have to create different types of cells. So the type of cell that I am working with is a mesenchymal stem cell. They make up skeletal tissue such as cartilage, and fat such as our buddy down here, and bone marrow, what's in these bones here. And the big thing that we focus on with MSCs are their natural anti-inflammatory factors. We want to be able to use that to reduce the inflammation. So, to be able to use them in therapies, we have to inject them into the body systemically, which means through an injection or through an IV. And with this comes the big problem with using them, is that they are really, really bad at listening to directions. So if we inject them into the body and we tell them and we want them to go to the hip to reduce the inflammation in the hip, they're not going to do that. Instead, they're going to do three things. They're either going to get stuck in the lungs or other filtering organs. They're going to attach to some place where we don't want them, and they're going to pass through the body. And this process is called pore homing. So how do we fix this? All cells are kind of like tennis balls. They have fuzz on the outside, or binding molecules that help them stick to other things. And so it's kind of like if you were to take a tennis ball and throw it up against a wall of Velcro, it will stick to that Velcro. Well, cells are similar in that the binding molecules on the outside of the cell will help them stick to other cells. And so the way that we can fix this is we can change those binding molecules that are expressed on the outside of the cell. We can make them more sticky or less sticky, and we do this through a process called small molecule treatment. And it's kind of similar to making somebody sneeze. If a person's just sitting in a room, they're not probably just normally sneezing over and over. However, if you take a dandelion and you rub it under their nose, they're probably going to sneeze. So we take these cells that aren't necessarily always super sticky or not sticky at all, and we'll treat them with another molecule that will make them sticky, or make them less sticky, and then we'll introduce those cells that have been treated into the body and see if it affects their homing to the site of inflammation to help reduce that inflammation. So we can see this here. Here we have a little mouse, and this ear is not inflamed, and this ear is. So hopefully we see that all of our MSCs are going to be going to the site of inflammation. But if we look at this picture, we can see that we have a lot more blue in this picture than we do have green, and that's what we want to see, because the blue are the modified cells and the green are just regular cells. And so all the cells that are blue here have been treated with another molecule that have helped them somewhat increase their homing. So we're trying to kind of build on this and maybe even get more cells to the site of inflammation. So my studies that I will be working towards in the future, and what I've started working on, has been treating the MSCs with a molecule called nitric oxide. Nitric oxide reduces the stickiness of the cell, but it also has shown to increase the lifespan. So the cell will be less likely to stick to places where we don't want it to stick, and it'll last longer in the body, so it'll hopefully reach the site of inflammation. So far this semester, I have learned all of my background research on MSCs and nitric oxide, and I'm learning about cell culture, which is raising and growing your own cells. I've read research papers on it, and so far I've conducted 2 experiments. Out of all of this, the hardest part has been reading the research papers. Unless you're an expert in the field, they're basically written in a different language. So, all of the work with nitric oxide and the mesenchymal stem cells is kind of my focus, that's my goal. But before I could start doing that research, my lab and my advisor gave me a project to create a cell growth curve, because they wanted to be able to see how well I can go through the scientific process and test hypotheses and plan an experiment. So they gave me the assignment to create a cell growth curve of the MSCs that we work with. So this is a cell growth curve. It shows the cell growth over time, and it has 4 distinct phases. It has the lag phase, which is right after you seed them into their growing places. They're going to take a while to get used to where they're going, or where they are. And then they're going to grow exponentially. They're going to start multiplying. And then after a while, they'll slow down and then they'll die off. There's 2 ways that we can check on our cell growth. There's the actual number of cells that we have, and we'll obtain that through a cell counting procedure, or we can observe the confluency. And the confluency is the percentage of the growing surface that we look at is covered by cells. And so here we can see that we have our flask that our cells grow in. You can see the cells stuck on the bottom. And when we look at that under a microscope, it'll look like this. And so from this picture, we can see that about 10% of the growing surface that we're looking at is covered in cells. Here it's about 30, 50, and 90. And so we'll observe that and kind of get a ballpark estimate of how many cells we have and just kind of look at how healthy they are. So this is a quick video of me going through the cell counting procedure. And this is how we obtain the actual, or the closer estimate of how many cells we actually have. So the cells reside in this little box here. It's kind of like a refrigerator, but it's called an incubator. And it keeps the cells at 37 degrees Celsius, which is body temperature, and it mimics the humidity and other factors that the cells would have in the body. So it's basically mimicking the environment that the cells would normally have. And so now I take the cells out, and now I'll observe the confluency and kind of get an idea of how the cells are. So I'll look under the microscope and record their confluency, and hopefully we'll be able to see up here on this screen, I'm not sure how good the resolution is, some blobs up on the screen, and these will be the cells. So yeah, we can see a few blobs here and there. Those are the cells. So this is a very low confluency of what I'm looking at. So once we observe our cells and we record our confluency, we're going to get ready to actually get our cell count. So what we do is we use the liquid called trypsin right here, and we will release the cells from the bottom of the cell flask in which they're growing in, and they'll come up into the liquid that they live in called media, and we will, from that, we'll be able to go through a process and figure out how many cells we have. So what we do all of this in is called the hood. This is a completely sterile environment. It doesn't allow any bacteria in. So before we do anything, we spray everything down with 70% ethanol to kill all bacteria, because we don't want any contaminants getting into our cell culture. So once I get all of our media warmed up to body temperature, because we don't want to shock ourselves, we will bring it into the hood, and we'll go ahead and start releasing the cells and getting them into a solution. So here, I will treat the cells with the trypsin and release them from the bottom of the flask. I will then put them in a vial and spin them down super, super fast, and it will create a solid cell pellet. And all my cells will be in that pellet, and then I will resuspend them in a controlled amount of liquid. So I know that I have all of my cells in, say, 1 milliliter of media. And from that, I can put it in a device. I can put a very, very small amount of that solution into a device called a hemocytometer. And that hemocytometer will allow me to count just a fraction of those cells I have. And so then I will put those counts that I get into this spreadsheet, which is mimicking an equation that we use to get an estimate. So I'll take 2 counts, it'll average them out, and then I'll put in how much media or fluid they're suspended in, and it'll give me an estimate of how many cells I actually have. So if I put 100, if I were to count 100 twice, then in the end I would have 1,500,000 cells. So that is how we obtain the estimate of how many cells we have, and we'll do that every day, and hopefully, and it will create a curve like this, hopefully. So as I said earlier, we have the 4 phases. Now this whole process takes over a week, and I only ran my experiment for about 5 days. So all we're going to observe in my results are these first 2 phases, the lag and the log phase. So these are my actual results. As we can see here, we have a very distinct lag phase, and then it starts to climb and starts to grow exponentially eventually. So from this, we can see that my cells did show to follow the expected trace. So now we're gonna look at the second way to estimate our cell count. We have the confluency, and so the optimal confluency for your cells is about 75%. That's when they're healthiest, and that's when you want to use them for experiments. So from this experiment, we can figure out how long it takes for our cells to get to their optimal confluency, when we want to use them. So from the data, we can see that on day 3, our cells were averaged out at 73% confluency, which is the closest data point to 75. So we can say that it takes about 3 days to get to optimal confluency. So our conclusions are that the cells did show to follow the predicted phases, and the estimated time for optimal confluency is about 3 days. And so this is the research that I have done. I'll be working in the future towards the nitric oxide. And are there any questions? Yes.
What was your favorite part of your internship this semester?
Definitely my advisor. No, it was probably learning the cell culture. I like, being in the hood and working with the cells and things like that. It was just, the hands-on portion is probably my favorite.
So what do you think will be next semester? What do you think?
So next semester I'll be continuing my research with nitric oxide. Actually, last week I started my first experiment with it, just kind of testing the waters, and so I'll start really diving into the nitric oxide experiments, testing the effects on the homing of the MSCs once they've been treated with the nitric oxide. Yes.
For the treatment of the nitric oxide, do you just bubble in NO gas with into the media, or how is that?
So it is. It's in forget the term, but it's in a powder that we use, and then we put it in there, and after a while of culture, it's then released into the flask that the cells are in, and so then they'll be exposed to it.
All right, good job.